Good morning. Thank you for joining us today. It is my distinct pleasure to introduce Professor Ravi Bhatia. Dr. Bhatia joined the University of Alabama at Birmingham in early 2015 as professor in the Department of Medicine. He's director of the Division of Hematology and Oncology and deputy director of the UAB Comprehensive Cancer Center. He received his medical training from the All India Institute of Medical Science in New Delhi, and his hematology, oncology, and bone marrow transplant training at the University of Minnesota, where he also completed a postdoctoral fellowship in stem cell research. In 1996, Dr. Bhatia joined the Department of Hematology and Hematopoietic Cell Transplantation at the City of Hope, and there he developed his career as an internationally recognized physician scientist. He served as professor and director of the Division of Hematopoietic Stem Cell and Leukemia Research and was co-leader of the Hematopoietic Malignancies program there. Dr. Bhatia's work has advanced our understanding of the biology and targeting of leukemia stem cells. He characterized the molecular and cellular events which underpin abnormal cell behavior in CML and made the seminal observation that when you treat patients with CML patients with tyrosine kinase inhibitors, were unable to eliminate the leukemia stem cell. And this observation shifted the focus in CML research to development of measures to eliminate leukemia stem cells and enhance opportunities for cure. And he will discuss this further in his talk. Dr. Bhatia has authored over 120 publications in major journals. He has received grant funding, continuous grant funding from the NIH since 2006. He's a scholar of the Leukemia and Lymphoma Society, where he also serves now in their advisory board and grant review panel. And he's a charter member of the NIH Hematopoiesis Study Section. He's an elected member of the American Society of Clinical Investigation and the Association of American Physicians. Dr. John Cory was very excited for you to come and talk to us today. And without further ado, please welcome Dr. Bhatia.
Thank you very much for the very kind introduction. I was invited to come here by John Cory, and the first thing I wanted to do was to express my condolences for your loss, and I feel it like a personal loss as well because he was a good friend. He's when he went to meetings, he was always a friendly face. Someone who always appeared to, who was always, happy to see you, made you feel that he was happy to see you, engaged you. And, when I moved to Alabama 2 years ago, he was very excited because he said, now you're close and we can really start to collaborate and do work together. So I think it's a loss, to all of us. And he's the person who invited me, and it's really we're in a situation where he's not here. But I think one of John's passions was CML. So he really led the way in a lot of the therapeutic trials of TKI treatment in CML. So it's I think appropriate, to talk about it, today. And these are trials that, John took part in, and as you know, CML is a disease which is caused by the BCR Abel oncogene, which results from the Philadelphia chromosome, the 922 translocation. So we have found, and many others have found, CML to be a really good model in which to, study leukemia, biology, and targeted therapies. And even though it seems to be a simple malignancy, it's caused by a single gene, there's still a lot of complexities to it. And I think what we learned from CML will help us as we work with more complex malignancies, with more complex genetic background, such as AML, other MPNs and MDS. So what we know is that in CML, BCR-ABL tyrosine kinase inhibitors are highly effective in treatment. They induce remissions in most patients, and they will lead to marked prolongation of survival. However, there is one sort of caveat to the success of tyrosine kinase inhibitors, which have basically, are the frontline treatment of CML. And that is that, we and others have shown that if you take patients who are on tyrosine kinase inhibitor treatment and are in deep remission, you still have persistent leukemia stem cells. And this is work, from my lab. It's a very simple study. We took bone marrow from patients who were in deep remission after several years of tyrosine kinase inhibitor treatment. We flow sorted, CD34 positive, 38 positive committed progenitors and 38 negative primitive progenitors / stem cells, and we analyzed them for the BCR-ABL gene. And basically what you can see in blue is that we can see high levels of expression of BCR-ABL within the stem cell compartment as compared to more committed progenitors or the bulk mononuclear cells. And one can quantify these primitive leukemia cells by doing limiting dilution analysis. And you can see that about, 0.5 to 1% of the CD38 negative cells in patients who are in remission for, up to 7 years after treatment and who have undetectable minimal residual disease, may still have, are still BCR-ABL positive. So even though there is a depletion of leukemia cells and maybe even a depletion of leukemia stem cells, there is a population of leukemia stem cells that is not eliminated. And why is this important? Well, we found out that this was important because quite early on, we found that if we stopped TKI treatment in patients for a variety of reasons, whether it was because of side effects or patients were not compliant or there was a pregnancy, that the disease would rapidly come back in most patients. Now, as time has gone on and we have, sort of refined the treatment, we found that there's a subset of patients who achieve undetectable minimal residual disease, that is, who are RT-PCR negative, who are able to discontinue therapy and in whom the disease may not come back. And the molecular recurrence-free survival after imatinib discontinuation is roughly about 40% in this select group of patients. But this group of patients really represents around 10 to 15% of all CML patients, because it has to be patients who achieve a deep remission, maintain it for a long period of time, and on a continuous basis. And not all patients get to that stage. So this, but it does show that it is possible, at least in some patients, to reach a state that the disease does not recur on discontinuing TKI treatment. So this is obviously very encouraging. And one question is, how can we sort of enhance this so that this is an option that's available to a larger number of patients? Why is it that the disease does not recur? I mean, one question is the leukemia stem cell eliminated in these patients? And the answer is that it's probably not. And this is a study from Tim Hughes' group in Adelaide, Australia, in which they did a DNA PCR, which was 1 to 2 logs more sensitive than the regular RT-PCR that we use in the clinic. And the black triangles here represent basically samples which are BCR-ABL positive. And so these are patients who are in treatment-free remission for up to 5 years. And you can see that in all these patients, at some point or the other, there is evidence of BCR-ABL positive cells. So it suggests that we're not eliminating all BCR-ABL positive cells in these patients. So this raises the question as to why is it that there are BCR-ABL positive cells and the disease is not recurring. So one possibility is that there's heterogeneity of these cells. The fact that it's long-lived, really suggests that these are cells with long-lived potential and are potentially stem cells. One possibility is that there's some stem cells that can hang around for a while but are not leukemogenic. The other possibility, which I think we also favor, is that there may be host factors that can regulate the residual leukemia stem cells, and these may be immune factors or they may be microenvironmental regulation. And then we also know that the length of treatment is a factor in maintaining treatment-free remission, which suggests that the actual quantity of residual cells may be a factor. So we think the recurrence risk may be determined by the quantity of residual leukemia stem cells, the quality of the cells, and their leukemogenic regenerative capacity, as well as host factors. And this is important because it gives us some additional sort of opportunities to think about how we may regulate and target leukemia stem cells. Why do we need to target leukemia stem cells? Because most patients, tolerate TKIs quite well, and can stay on it for an extended period of time. I think the reason is that as long as the leukemia stem cells are there, there's a possibility of disease relapse or progression. So the rationale is to really enhance the possibility of treatment discontinuation as well as to reduce the risk of relapse. We know that non-compliance is a major issue in these patients. To me, in my clinic, as I see CML patients, is the thing that I worry about the most and that I talk to patients about the most. There is a risk of side effects. There are some, most patients do well, but they often have nagging side effects which affect their quality of life, including fatigue and cramps, etc. And with the second generation and third generation TKIs, there is really quite a high risk of vascular adverse events, and which contribute to morbidity and even mortality. For younger patients, younger women, teratogenicity is a issue. And then finally, there is still a significant financial burden from TKI treatment, and the treatment can cost $100,000 per patient per year. There, depending on the way insurance is going now, patients can have high co-payments, which are unaffordable. And finally, there's an increasing prevalence of CML because of our success in treating this disease and maintaining patients surviving for a longer period of time. So this was actually reported in the Washington Post, in which they sort of described the increasing prevalence of CML from 2010 to 2015. And the number of patients living with the disease has been growing and is expected to keep on growing, and there's going to be increased demand for the drugs. But the drug prices have not been coming down; they've actually been going up. Even for Gleevec, which was the first drug which was approved in 2001, we now see that the price has almost tripled since it's come, and this is the price per month. So the cost to both to society and to the individual patients is quite high. This may change as generics come in. It hasn't happened so far in the US, but if I talk to colleagues in Sweden, where Gleevec is generic and all forms of generic Gleevec are available, the cost of Gleevec has dropped down to about 3% of what it was before. So, but then we'll have to reconsider how we treat patients with second and third generation TKIs, because then the cost benefit analysis is going to change as well, and so those are still issues which are important. TKI treatment, why is it that it doesn't eliminate leukemia stem cells? So one possibility is that it's not able to hit its target in the stem cells. Stem cells are known to have altered transporters. There may be poor drug intake into the cell or export from the cell. But this does not appear to be the case, because if you expose leukemia stem cells to TKIs, whether it's imatinib, dasatinib, nilotinib, you can see that there's very effective inhibition of tyrosine kinase activity, suggesting that the drug is hitting its target in the cell. So this suggests that, and the concept has really been, I think, adopted, uh, very widely, that the survival of CML stem and progenitor cells after TKI treatment is not explained by lack of inhibition of BCR Abel kinase activity, and may represent BCR Abel kinase independent mechanisms. We also know that there is a population of stem cells which is very quiescent, which tends to escape elimination by TKI treatment. So this is a CFSC plot. As cells divide, the CFSC fluorescence reduces by half, and so as you move towards the left, these are dividing cells, and the cells which are on the right are the non-dividing cells. And you can see there's a population of non-dividing cells that escape elimination by TKI. And we know that non-dividing cells tend to be very resistant to all kinds of treatment. And lack of division is also a property of stem cells. So when we think of the potential mechanisms of persistence of CML stem cells, one can think that with TKI treatment is inhibition of BCR Abel kinase activity, but that there remain intrinsic factors as well as extrinsic factors that can act on the leukemia stem cells and which are able to maintain their survival. And these intrinsic factors may be induced by the BCR Abel gene, and but they may persist even after tyrosine kinase inhibition through epigenetic mechanisms or through regulatory loops which are able to sustain themselves after TKI inhibition. And we also know that there may be factors from the microenvironment which are important for normal stem cells, and there may be similar factors or different factors that are involved in regulation of leukemia stem cells. And for the rest of the talk, I'm going to discuss mostly work that we've been doing more recently related to the bone marrow microenvironment and how it might affect the leukemia stem cells that might be targeted. So we use a model of CML. In my lab, we study samples from patients because obviously that is the most relevant cell and tissue type to study. But to study mechanisms, we've also used a genetic mouse model of CML in which there is targeted BCR Abel expression in stem cells using an SCL promoter, which is regulated by tetracycline. So if you withdraw tetracycline from these mice, BCR Abel is expressed in stem cells, and the mice develop a CML-like disorder with leukocytosis, neutrophilia, infiltration with myeloid cells, splenomegaly, and the mice die, over a period of weeks to months. And we have shown, and others have shown, that in this model, as in patients with CML, the leukemia stem cell shares characteristics with normal stem cells, and it shares the exact same phenotype as the normal stem cell. So this fits in with the CML resulting from transformation of a very primitive stem cell by the BCR Abel gene. So one of the things that we found when we used this model was that the CML microenvironment seems to provide a competitive advantage to leukemia stem cells compared to normal stem cells. And so we took leukemia stem cells and we took control stem cells, and we injected them into leukemic recipients and control recipients, and we measured their growth. And essentially what we found was that control stem cells did not grow well. Normal stem cells did not grow well in leukemic mice, whereas they grew as well in leukemic mice, whereas leukemic stem cells grew as well in leukemic mice as they grow in control mice. So this suggests that in the leukemic microenvironment, the leukemia stem cell has a competitive advantage. And we can show the same with the bone marrow supernatants from or conditioned medium from leukemia bone marrow, where we can show that if we mix CML and normal stem cells, that there's a competitive growth advantage to the leukemia stem cells in the presence of CML conditioned medium. So we looked at the cytokine and chemokine profile in these mice using multiplex ELISA, and we found that there were multiple alterations in the expression of different cytokines in the bone marrow of leukemic mice. So CXCL12 is a very critical chemokine responsible for stem cell homing and localization to the niche. And that was actually reduced in leukemic bone marrow, whereas the levels of several other growth factors and inflammatory cytokines such as IL-1 alpha, beta, TNF alpha, IL-6, and MIP-1 alpha and beta, and many others are upregulated in the bone marrow of leukemic mice. These cytokines are derived variably from both the leukemic cells as well as from the non-hematopoietic bone marrow microenvironmental cells. So, and we believe that they play a potential role in regulating the differential growth of leukemic versus normal stem cells in the leukemic bone marrow. One of the factors that we've studied in more detail and reported on is interleukin 1. And we were interested in interleukin 1 because it's a pivotal pro-inflammatory cytokine. And there's also evidence from other groups and confirmed by us that the receptors for interleukin 1, which is IL-1 R1 and IL-1 RAP, which are the two co-receptors, are overexpressed in CML stem cells as compared to the normal counterparts. If you stimulate CML stem cells and as compared to normal stem cells with interleukin 1, you see enhanced responsiveness with increased NF-kappa B, MAP kinase, and JNK activation. And we used an agent called IL-1RA, which is a naturally occurring inhibitor of IL-1 activity. And we found that if we treated mice with nilotinib, IL-1RA, or the combination, that the combination resulted in enhanced inhibition of leukemia stem cells compared to nilotinib alone. And that if you stopped nilotinib and IL-1RA and followed mice for survival, the mice that had received IL-1RA survived longer than mice that had received nilotinib alone. So this suggested that there was some activity of the combination against leukemia stem cells. We confirmed these findings with the human stem cells, and you can see CML CD34 positive, 38 negative progenitors are inhibited by the combination in terms of cell number, cell proliferation, the CFSC assay, and colony forming assays, and also show increased apoptosis. So we've also showed that when we treat with nilotinib and IL-1RA, that we see enhanced inhibition of NF-kappa B and P38 in JNK as compared to nilotinib alone or IL-1RA alone. And we confirmed this using immunofluorescence, where you can see that there's increased inhibition of nuclear NF-kappa B with the combination. So the model that we devised from this, which we think is active maybe for other cytokines as well, is that in CML, we know that BCR Abel can directly, through intracellular signaling mechanisms, activate NF-kappa B, and this is a feature of CML. But we also think that the microenvironment, through the production of inflammatory cytokines, increased expression of the cytokine receptors, can lead to NF-kappa B activation. And NF-kappa B activation itself can lead to increased expression of inflammatory cytokines in addition to its effects on growth of stem cells. And so we think that when we treat with TKIs, we are blocking the BCR Abel induced activation of NF-kappa B, but the microenvironmental activation is still active. And that if we treat with the combination, we're now blocking both the mechanisms of activation of this signal, and that's why we may be seeing an enhanced effect. So IL-1RA is available for the treatment of rheumatological conditions, including rheumatoid arthritis and JRA, and it is a possible approach to targeting leukemia stem cells in combination with Gleevec in the future, or in combination with other TKIs in the future. So we're also interested in understanding more about the nature of the changes in the microenvironment in leukemia. We know that the bone marrow microenvironment is very important for the regulation of normal hematopoietic stem cells. And there's been a lot of work done over the last 10 years or so, which has really helped to characterize the cells within the microenvironment that might have a role in stem cell regulation. And they're shown here. And they include both hematopoietic cells and non-hematopoietic cells. We think that cells associated with the vasculature, including endothelial cells, as well as perivascular cells, which are thought to be mesenchymal stem cells, may play a role, appear to play an important role in regulation of normal hematopoiesis. There's also some evidence that cells associated with the bone may play an important role, as well as there's evidence that neuronal cells may contribute as well. In addition, we know that there's a lot of interest in the role of megakaryocytes and macrophages in regulating normal hematopoietic stem cells. And we know from our studies looking at the cytokines that the differentiating leukemic cells can produce factors that may affect leukemia stem cells. So the bone marrow microenvironmental components regulate stem cell quiescence, self-renewal, and differentiation. And the questions are, do leukemic cells really alter the distribution and function of the non-hematopoietic bone marrow microenvironmental cells? And what is the nature of the leukemic stem cell niche as compared, is it the same niche as is used for normal hematopoietic stem cells, or is niche function altered, or do leukemia stem cells use a different niche from normal hematopoietic stem cells? And this is very important if we're looking to target stem cell and niche interactions. So we've looked at this in many ways. Hematopoiesis is very well characterized by surface markers. The mesenchymal compartment is less well characterized. But recently there was a paper from Longecker and Weissman in Stanford in which they described a murine bone marrow skeletal stem cell hierarchy with a mouse skeletal stem cell, and then individual lineage related progenitors, including a cartilage progenitor, a bone progenitor, and a stroma progenitor. And we focused on the stem cell, the bone progenitor, which is called thigh cells, and the stroma progenitor, which is called 6C3 cells. And we looked at this in the leukemic mice, and we found that there was increased expression of the bone-forming increased numbers of the bone-forming progenitors and the stromal progenitors in the leukemic mice. But we're more interested in the function of these progenitors, and to evaluate this, we took both normal and leukemic stem cells and co-cultured them with normal or leukemic skeletal stem cell or skeletal progenitors, and then took the cells after co-culture and injected them into mice to evaluate the effect on engraftment and stem cell function. And so what we found was that for normal hematopoiesis, that the normal skeletal stem cells were quite effective in maintaining normal stem cells, and to a lesser extent, the skeletal and the stromal progenitors also maintained normal stem cells. On the other hand, the CML skeletal stem cells did not support normal hematopoiesis as as the normal counterparts did. And this fits in with what we saw in vivo as well when we injected normal stem cells into leukemic mice. On the other hand, when we looked at CML stem cells, we found that the CML stem cells were not really supported by the normal skeletal stem cells or by the leukemic skeletal stem cells, but there was increased support of CML stem cells by CML stroma as compared to normal stroma. So this suggested a model in which there may be increased support of leukemia stem cells by CML stromal cells, but CML stromal cells do not support normal stem cells as well, and this could give a competitive growth advantage to the leukemia stem cells. We're in the process of doing RNA-seq, but prior to that, we've done some fluidime analyses and look at some of the common genes which are known to affect hematopoietic regulation and may be expressed by the bone marrow microenvironment. And we find that compared to normal skeletal stem cells, the CML skeletal stem cells show increased expression of some of these growth factors and cytokines, and reduced expression of factors which are known to be important for regulation of normal hematopoiesis or normal stem cells, including CXCL12, which I mentioned before, integrin alpha 4, MIP1 alpha, and frizzled receptor. The CML thigh cells also show some reduced expression of CXCL12 and ROBO4, which are important for normal hematopoiesis. And the CML63 cells, interestingly, show increased expression of a number of Wnt ligands, and suggesting that this may be a potential mechanism by which the CML stromal cells demonstrate enhanced support of leukemia stem cell growth. And in summary, what we found is significant alterations in the distribution and function of skeletal stem cell subsets in CML bone marrow. That in normal hematopoiesis, the skeletal stem cells support normal hematopoiesis, and the CML skeletal stem cells and progenitor cells show reduced support of normal hematopoiesis. And that, on the other hand, the CML stromal cells show increased support of CML hematopoiesis. This may have some implications for targeting, and we've recently again published that there is increased expression of a few of the frizzled receptors, which are the receptors for the Wnt ligands on CML CD34 positive cells. So frizzled 4 and frizzled 5 are expressed at significantly higher levels on CML CD34 positive cells as compared to the normal counterparts. And we also know that if we expose CML cells to Wints, that they show increased colony formation, and that and that knockdown of Wints leads to inhibition of colony formation, and in combination with nilotinib leads to even further inhibition of colony formation. And we used an inhibitor of Wnt secretion. This is Wints are secreted. Wnt secretion requires palmotylation of the Wints by an enzyme called porcupine, and the palmotylation is required for the secretion of activated Wnt and for Wnt activity. And if you use a porcupine inhibitor, this is LGK974, you can see that you can inhibit Wnt signaling, and you can also see that there is reduced expression of Wnt target genes in CML CD34 positive cells. And we've used the porcupine inhibitor in functional assays and show that the inhibitor affects the proliferation of CML stem cells and the enhanced activity in combination with nilotinib. It reduces the growth of CML stem cells in an immunodeficient mouse model. This is a xenograft model of CML where we can see significant inhibition using the combination of nilotinib and the porcupine inhibitor. We've treated the genetic mouse model with these inhibitors and shown again that the combination shows increased activity over nilotinib alone, and that if we treat and then discontinue treatment, that there's enhanced survival after discontinuation of treatment. So this suggests that targeting Wnt inhibition may be another approach that could be helpful in inhibiting and depleting leukemia stem cells in CML. The Wnt inhibitor is currently in phase 1 trials in a variety of solid tumors, and we're awaiting to see whether there's opportunities for further development of this inhibitor. And then the last thing that I'll talk about is some other characterization of the nature of the leukemia stem cell niche. So the studies that I showed you prior to this were done in vitro. We identified cells, we isolated them, we cultured in vitro. We wanted to do some in vivo studies to see really what was the function in the in vivo setting. And we used a model in which you can delete CXCL12 from a variety of bone marrow microenvironmental cells using the CRE-LOX system. So with the CRE-LOX system, you could target the CRE enzyme to a variety of different cells using different promoters. So if we use, for example, the PRX promoter, the CRE is expressed in mesenchymal stem cells. If we use the Ostrix promoter, the CRE is expressed in osteoprogenitors. If we use a TEC CRE, it's targeted in endothelial cells. And if we use the osteocalcin CRE, it's targeted to osteoblasts. So the reason to do this is that we know that CXCL12 is a critical chemokine for normal hematopoiesis, that if you, and it's required for stem cells to localize to their niches. And what's been shown is that if you delete CXCL12 from PRX expressing cells and to a lesser extent from, which is from mesenchymal stem cells, And if you delete CXCL12 from endothelial cells, you see a marked reduction in normal hematopoiesis, suggesting that the CXCL12 expressing mesenchymal stem cells or endothelial cells have critical niche function for normal stem cells. So what happens in CML? So to answer this question, we basically took CML, we basically took these different Cree mice in which we've knocked CXCL12 from different populations, including endothelial cells, mesenchymal stem cells, osteoblasts, and osteoprogenitors. And we injected normal or CML bone marrow into these mice and evaluated the growth in these conditions. And we confirmed what was described before for normal LTHSC, where we saw a marked reduction in the number of LTHSCs that were regenerated within the mice, in which CXCL12 was deleted from mesenchymal stem cells using PRX1-CRE. We did not see a significant effect of Ostrix or osteocalcin-CRE, or even with the TEC-CRE, which deletes it from endothelial cells, which is a little bit different from what was described before. However, when we looked at CML, we found that it was markedly different. And if you remember, in the normal stem cells, PRX-CRE, deletion of CXCL12 from PRX, using PRX-CRE led to a marked depletion of stem cells. In CML, it actually led to enhanced leukemia development, increased bone marrow cellularity, increased LTHSC numbers, and actually reduced survival of the mice. On the other hand, if we looked at TEC-CRE, it didn't have a significant effect on normal hematopoiesis. It didn't affect WBC, but it did lead to reduced bone marrow cellularity and reduction in LTHSC numbers, and it led to some increase in survival of mice. So this suggested that the deletion of CXCL12 from MSCs enhances CML development, whereas deletion from endothelial cell depletes LSCs and reduces leukemia development. We confirmed that this was affecting stem cells by doing secondary transplant, and we show that cells from PRX-CRE mice show enhanced secondary repopulation, whereas cells from the TEC-CRE mice, where CXCL12 is depleted from endothelium, showed reduced donor engraftment. We also found that in CML that the number of CXCL12 expressing endothelial cells was increased, and we used a CXCL12 GFP mouse, which we crossed with the BCR Abel mouse, and and we could show that the expression of BCR Abel led to an increase in CXCL12 expressing endothelial cells, and it led to a reduction in CXCL12 expressing mesenchymal cells. So these are the cells which, when they're and we've showed before that depletion of CXCL12 from mesenchymal cells enhances CML. So we could see that the reduction of CXCL12 expressing mesenchymal cells may play a promotional role in CML. And we've shown that endothelial cells actually enhance CML, and so the increase in endothelial cells may again, be a factor that leads to enhanced growth of leukemia. So the model that we have is that in normal hematopoiesis, we have a situation in which the normal hematopoietic stem cells are more dependent on CXCL12 expressing mesenchymal stem cells, whereas the leukemia stem cells seem to be more dependent on endothelial cells. When you delete CXCL12 from the mesenchymal stem cells, there's a loss of normal hematopoiesis, and it allows an increased growth or a competitive overgrowth of leukemia stem cells. On the other hand, if you delete CXCL12 from endothelial cells, you're losing, the niche for leukemia stem cells, and but you're not affecting hematopoietic stem cells. So this, raises the possibility of further characterizing the interaction between endothelial cells and the CXCL and the mesenchymal stem cells and leukemia stem cells as a means for subsequent improved understanding of regulation of leukemia stem cells and possibly for improved targeting of these cells. We also think that these alterations may, as I mentioned, play a role in CML evolution, and that in the normal bone marrow niche, the PRX1 positive mesenchymal stem cells expressing CXCL12 are supporting HSC, and that with the development of leukemia, there's a reduction in these cells, and that leads to a disadvantage to HSCs, where there's an increase in the number of CXCL12 expressing endothelial cells, which provides an advantage to the leukemia stem cells. So I think broadly the conclusions from this section is that we think that there is bidirectional regulation. The bone marrow microenvironment clearly regulates both CML and normal stem cells growth and drug response. On the other hand, we also know that the leukemia induces changes in the microenvironment, which alter the regulation of both the leukemic and the normal hematopoietic stem cells. And I think that improved knowledge of niche regulatory mechanisms has the promise not only to yield improved understanding of leukemia evolution, drug response, drug resistance, and disease persistence, but may also guide development of better approaches to target CML leukemia stem cells, which I think is something that we've talked about for a long time and which we'd really like to see some more progress with. And just to give you an idea, there are many people who are working in this area, and there are many possible mechanisms. And as I'd mentioned before, these could be both external mechanisms as well as intrinsic mechanisms. And several of these mechanisms are actually being tested in different clinical trials. So there are clinical trials that we had using an HDAC inhibitor. Interferon has been used. Chloroquine is being used to enhance as an autophagy inhibitor. There is smoothened inhibitors have been tried. Pioglitazone as a PPAR gamma activator is under trials with the French, and the trials of ruxolitinib, which are a couple of trials of ruxolitinib which are underway. So there's a variety of possible mechanisms which are being evaluated. And, I like to think of this as being, an old Indian fable of the 6 wise men and the elephant. And these were all blind men, and they're feeling different parts of the elephant. And, and depending on which part of the elephant they're touching, they're giving their own interpretation of what is happening. And I think, we all have our own favorites and our own biases. And what's going to be critical is to sort of build a combined system sort of level view of what are the critical mechanisms for retention and maintenance of leukemia stem cells, and to try and identify what is critically different between those mechanisms and mechanisms active in normal stem cells to be more successful. So I'd like to thank all the people in my lab who are responsible for doing this work, and also for people who worked with me while I was at City of Hope, and for the different funding agencies. Well, thank you very much.